Yeah, thanks a lot for the introduction. I'm Lorenz, working together with Ben Engel in Basel and also Tingying Peng in Munich. And yeah, thanks also for the chance to present our project here, which is Membrane V2. And I would like to start with a small motivation of how we actually started this project. And that was work together with Ben Engel and Wojciech here, where we tried to learn a bit more about photosynthesis. So we were looking at different organisms, for example, this Chlamydomonas cell here, which performs photosynthesis. And it does that within the thylakoid membranes here, which can be organized in very specific ways. And depending on their organization, also their functionality changes. So we want to study what's going on there. But at the same time, we also want to look at this single complexes scale, which means the proteins that are embedded in the membranes. In this case, that's the photosystem 2 proteins that capture the energy of light and start the reaction of photosynthesis. And they can be organized in very specific ways too. And depending on their organization of the membranes, also the organization of the proteins changes. So we want to study both of these scales at the same time. And to be able to do that in a larger scale, we need to have accurate membrane segmentations. And we need to know where the proteins are. So we require the protein locations. And this is exactly what we developed MemBrain for. So MemBrain is our tool for the analysis of membranes, which will consist of 3 different components. The first one is MemBrain-seg, which is already publicly available. That's our component for membrane segmentations out of the box. So if you haven't tried it yet, feel free to give it a try. The next component is MemBrain-pick, which should be a very interactive tool for the localization of proteins. And the third thing is MemBrain-stats, which basically takes the outputs of the first 2 steps and automatically outputs some statistics. But today I would like to focus on the first 2 components here, MemBrain-seg and MemBrain-pick. So let's start with MemBrain-seg. So that was a really nice collaboration with Alistair also and Kevin. And our goal with that was to get some pretty good out-of-the-box segmentations without user requirement to annotate anything or fine-tune anything. So you can see it's working nicely on many tomograms. But at the same time, we cannot guarantee that it's working for everybody. So we also want to make it easy for the user to just annotate a couple of patches and then fine-tune on their data to improve the performance here. And yeah, once these patches are annotated, we also, of course, encourage users to share them with the community so everybody can benefit from these annotations. How did we actually design MemBrain-seg? Yeah, we used a unit architecture, which is, I think, still the architecture to beat for medical image segmentation. And very importantly, we also tried to get a very large variety into our training data set, getting a lot of variation in how membranes can actually look. But at the same time, we also tried to artificially increase this variability by some strong data augmentations to increase how membranes can actually look. And yeah, these 2 points down here, they are maybe not the most exciting design choices of a neural network, but I think they are very important. And that's why I would like to focus a bit more on how we actually did this. So for the generation of the training data set, we followed a very iterative approach. So we started with tomosegmemTV, which gave us some reasonable membrane segmentations, but they were not as good as we would have liked them. So we manually corrected them and trained a first round of MemBrain-seg. Then we again predicted on some tomograms, extracted regions where the performance was bad, corrected again, and retrained. And iteratively, we got better and better and increased our training data set with that. So Simon up here, he did a great job with that. And one thing I want to mention here is that we just focused on small patches. So we just extracted these patches of size 160. And the reason being that I think you cannot really correct a whole tomogram and make sure that all the annotations are correct there. But for these small patches, we really try to make sure that each voxel is basically assigned to the correct class. So this looked something like that. So on the left side, you can see a prediction of some iteration of MemBrain-seg. And on the right side, you can see our correction that we then also use for our training data. And what we did was that we painted in some areas where we saw, okay, there should be a membrane. We also removed some false positives. And very importantly, we also assigned this ignore label. It's the green label here. So we always assigned that whenever we were not really sure what was happening. For example, if the missing wedge removed some densities, or we were just not sure whether there is a membrane or not. And what happened then was that during training, these green voxels, the ignore label voxels, they are just not evaluated by the loss function, and they will not distort the training in either direction. And with that approach, we got a nice training data set, but as I said, we also try to artificially increase the variability and also the size of our training data set using some data augmentations. Here are some very classical data augmentations that we included into our training, but at the same time, we also came up with some custom augmentations that are maybe more specific to cryo-ET or also to membrane segmentation, and which rely more on corrupting the Fourier space of the patches. So here you can see the patch together with its Fourier transform. And the first thing that we did was this missing wedge transformation, where we just randomly crop out a missing wedge in Fourier space, leading to also some densities in real space obviously disappearing. So there is this membrane which you can see in the original patch, but after this missing wedge transform, you cannot see it anymore. But because we know from the original patch that there should be a membrane, we still have the ground truth for that, so we can try to train the network to still fill in these areas, judging by the context. So in order to try to kind of mitigate the effects of the missing wedge to some extent. Another thing that we did was this Fourier amplitude transformation, where we just randomly rescale the intensities of some Fourier frequencies. In this case, we upscaled the lower frequencies, leading to the lower frequency features, so the coarser features being more pronounced and the membranes also being much more visible. But we also did the opposite thing, basically, where we just upscaled the higher frequencies, leading to the image being much more noisy. So this is basically the data that we presented our network during training. But then also the question is, how do we actually optimize it? And as a loss function, also for evaluation, the natural choice is the dye score. So here you can see a prediction of membrane compared to the ground truth. And if we compute the dye score, that's 80%, which I think is a reasonable score. One thing that we observed, though, was that depending on who is annotating the patches, also the thickness of the annotations changes a lot. So depending on the brush size you choose in your annotation software, for example. And what happens with the dye score is that it gets increasingly worse. So we wanted to mitigate that by coming up with a metric that doesn't really focus too much on the pixel-wise agreement, but rather focuses on the connectivity of the membranes. So we developed the surface dyes, which is largely also inspired by the centerline dyes, if you're maybe familiar with that. And here we are really not trying to compare the prediction with the ground truth on a pixel level, but we instead skeletonize both the prediction and the ground truth, and then we see what fractions of the prediction and the ground truth are actually correct. And because these predictions, because these skeletons, they are invariant basically to the thickness of the annotation, right? We always shrink them down to a 1 voxel thick sheet, no matter how thick they are in the beginning, because this is invariant to the thickness of the membrane. Also, our surface dice is invariant to the thickness of the membrane. And this is nice because we can use that for evaluation, but we can also at the same time use it for training our model and to focus more on the connectivity of the membranes. And that already brings me to the summary of MemBrain-seg. So it works pretty well out of the box. We also have some quantitative evaluations in our preprint if you want to check that out, where we also showed how the different components that I just showed you also contribute to our model performance. And one thing that I also want to mention here is that it's very easy to use. So it's a Python package. You can just install it via pip install MemBrain-seg, and then it's a single command line getting some segmentations out of your tomograms. Now, in the remaining time, I would like to focus on MemBrain-pick, so our membrane protein localization pipeline here. And why do we actually need this? So in the first place, we did some benchmarking here. So we gathered some ground truth data for a couple of membranes where we tried to find the best working method for this. And of course, we tried template matching, which didn't perform so well. So we're looking at we want to have as many white squares as possible. So that was not really satisfying, and the same also for a couple of deep learning methods that we tried, Priolo, DeepFinder, and EMAN2. So I think 2 years ago, we came up with MemBrain, which was back then V1, and that was performing already pretty nicely. So the benefits of MemBrain V1 was that it got a pretty decent performance already with very few annotated membranes. So it was very annotation efficient. So we didn't need a very large training set to perform well. The problem was that the performance was still improvable, I think. So we still spent quite some time correcting the picks of MemBrain-pick. And I think the reason was that it just didn't consider a lot of context. So it was just extracting some small subvolumes on the membrane, and that was the entire receptive field, which was very, very low, very small. So this is something that we want to improve now for MemBrain-pick. So we want to get a better performance, but also incorporating a bit more context in the membrane. But at the same time, we also want to keep this annotation efficiency because we don't want to rely on large training data sets, obviously. So we want to make this a very interactive approach where the user can go back and forth between annotation and training of the model. And how do we do this? So we want to learn directly on mesh representations of our membranes and then integrate that also with some proper annotation software. Now, the workflow of MemBrain-pick is this. So we start with this with a membrane segmentation. So that's just a binary segmentation, 0-1 voxelized segmentation. And we convert that to a mesh. So basically a surface consisting of many small triangles. And here you can see the vertices of the triangles. And what we will do then is feed this mesh here into a neural network. But before that, we also assign some features to each of the vertices. And we do that by projecting the tomogram densities along the normal vector of the membrane onto the membrane. So here you can see really the densities when going in and out of the membrane. So this is very similar to also Membrainorama, if you're familiar with that. And so what we do with that is then that we feed this into a neural network. What we want to get out is these ground truth positions that I visualized here in magenta over there. And to be able to do that, we first encode that into a distance map. So this is our training target, where we predict basically the distance to the nearest protein center. And as an architecture, what we do is that we choose a diffusion net, which takes as input a mesh and gets here as an output also a mesh together with some scalar values associated to the vertices. And the name diffusion net comes from this diffusion operation. So it's based on the diffusion equation, and it uses the diffusion to basically for the vertices to communicate with its neighbors. And there it has this learnable parameter, the time, which basically controls how far information can spread. And the nice thing about this is then that we don't need to define how much context the network actually considers, but it actually learns the range of context that is beneficial for the performance. So this is how we train the model. And then for inference, what we do, we feed in such a membrane here, and we get out a heat map, something like that. And you can already pretty well see where the protein should be. So what we do is just some mean shift clustering to get out some positions where proteins could be. And if we compare that to some ground truth positions, the one that I showed up there, then basically all of the ground truth positions are captured. There are some positions more predicted where I think there could be something, and that is something that we would need to look into here. Now, you may ask how to actually get to some annotations easily so that I can train MemBrain-seg. And that's a good question, because very recently we also had this hackathon in Basel together with Ricardo, Kyle, Lorenzo, and Kevin, where one of the goals of this hackathon was to make Membrainorama functionalities available in Surforama. So Membrainorama, that's this very nice annotation software by Dimitri Tegunov, where also densities of the tomogram are projected onto the membrane surface. And then you can click and annotate a couple of points here. The problem with it is that it's living in Windows, and it's also not so easy to integrate that into our Python framework. So we tried to make this accessible also in Apari, and that's what we did. So now there's this Surforama tool available which also projects densities onto your mesh, and you can even view the densities here in context. And then you can just start annotating and click a couple of points to get some nice annotations too. Then in the end, hopefully soon we'll be able to train MemBrain-pick. And that brings me to my summary slide already. So MemBrain-seg is working already pretty nicely. Feel free to give this a try. And MemBrain-pick, that's a work in progress, but I hope that it will be out very, very soon. I haven't talked too much about MemBrain-stats yet. So what MemBrain-stats will be doing is really taking the outputs of MemBrain-seg and MemBrain-pick and give you some automated statistics here. But I also want to mention that we don't want to reinvent the wheel here. So there are some really good packages out there. So it's also important for us to just make our outputs very compatible so they can be easily used in these other frameworks. And with that, I would like to thank everybody in the Peng Lab and also the Cell Architecture Lab and everybody who contributed to this project. And thanks to you for your attention.